Hello dear students, welcome back. This is Dr. Hadi. Today's topic is the structure and types of RNA. Of course, the chapter is nucleic acid. Today's topic will be we will exert more emphasis on the types of RNA rather than structure of RNA, because the structure of RNA has already been discussed in the structure of the DNA. Because if you know the structure of DNA, then you can suppose that you have you got the structure of the RNA in advance. Fine. The only difference between the DNA and RNA was that in the DNA we found usually there are two strands, but in the RNA generally there is only one strand in the RNA. And in the DNA the sugar is deoxyribose, while in RNA the sugar is ribose sugar, and this was the difference between the RNA and DNA. Now if I write here just a single diagram like this is the 5' and this is the 3'. It is a single strand. You can see there are bases, means you can see adenine, guanine, cytosine, but when the word thymine comes, then in place of thymine you will find uracil only. Fine. So you can go on. This will be the structure of the RNA, and RNA looks like DNA. RNA is single-stranded while DNA is double-stranded. Now the main topic is the types of the RNA. There are three types of RNA. I suppose I am expecting that you might have seen my video lecture on the nucleic acid. You can find the playlist as well. So the types of RNA there are three types of RNA. The one is messenger RNA, second one is the transfer RNA, and the ribosomal RNA. We will start now from messenger RNA. The percentage of the messenger RNA is from 5 to 10%. The very first thing, if you go inside a cell, you will find RNA, and then you will find the percentage of each type of RNA. So the first thing, that messenger RNA is 5 to 10% inside your cell. That messenger RNA, whenever it is synthesized, of course it is synthesized from the DNA. When it is synthesized, in the beginning it will be given some special structures on its 5' and on its 3', just for the sake of protection and stability. So it means that at 5' we will add a compound called 7-methyl. M stands for methyl. 7-methyl, that is methyl guanosine triphosphate. This is a compound. I will represent that compound with this diagram only. Fine. Okay, this is the diagram. Okay, and this diagram represents 7-methyl guanosine triphosphate. You can call it the cape, and some students consider it the real cape. It is not the real cape. It is actually that compound which is added to the 5' of the messenger RNA. Now it is called 7-methyl guanosine triphosphate. What is the function of this 7-methyl guanosine triphosphate? The function is protection and recognition. You can write here, one is the protection, okay, protection, and second one is the recognition. The messenger RNA must be recognized during protein synthesis, because all of these three types of RNAs, these are involved in protein synthesis, and in protein synthesis you need some sort of recognition. So messenger RNA can be recognized by its 5'. And protection, what about protection? There are some enzyme called the nucleases enzyme. These nuclease enzyme can break your messenger RNA. They can lyse your messenger RNA. So in order to prevent from these hydrolytic effects of the enzyme, we need this cape. It will protect from the breakdown process. Now at the 3' you will see another another compound, but a combination of compounds means many adenosine residues. Adenine nucleotide, you know very well adenine nucleotide. So many adenine, okay, from 20 to 250 new adenylate nucleotides will be added here. So now I will write a diagram like this. Fine. So many adenine. I think you know adenine very well. Adenine, guanine, cytosine. These are the nitrogenous bases which are present in the nucleic acid. So here many adenine molecules will be added at the 3 end. This is called as poly A tail. We can call it poly A tail. Poly A tail. Now what is the purpose of this poly A tail? Again, it provides what? It provides stability. It provides stability because the messenger RNA is a kind of a harsh environment. There is a chance that the messenger RNA is destabilized, means some changes occur in its structure. In order to save the structure of the messenger RNA, we need a poly A tail. It will stabilize that messenger RNA throughout its journey, because this messenger RNA is now supposed to go towards the ribosome, attach there, and the process of protein synthesis will start with the attachment of messenger RNA to the ribosome. Okay, so here I'm going to write the diagram. If I write the diagram here, let's suppose this is the ribosome structure, smaller unit, and this is the larger unit of the eukaryotic ribosome. I will write the ribosome. Okay, now I can change my marker. Now here you can see that messenger RNA will come here. This is the messenger RNA. Okay, this is the messenger RNA. This will be the 5' and this will be the 3'. Okay, so the messenger RNA once attached with ribosome, with the help of this messenger RNA, you will see protein synthesis. Means many amino acid will aggregate here. Many amino acid, 1 amino acid, 2, 3, 4, 5, 6, 7, 8, 9, 10, 120. So on the surface of the ribosome, you will see that many amino acids will join together and they will form a chain of shorter length or longer length, depend upon the condition and the situation. So the process is called protein synthesis. And for protein synthesis, the very first RNA was the messenger RNA. Fine. So here you saw the importance of the messenger RNA. And I remember one thing more, that a messenger RNA you will find lots of nucleotide like A, G, C, U, C. Okay, fine. So any 3 nucleotide, a set of any 3 nucleotides on the messenger RNA is called codon. It's called what? Codon. So here, if you see here, there is adenine, here cytosine, and again adenine. So ACA, this is called as codon. This is called as codon. And this codon represent a code that it has derived from the DNA. Fine. So we call it a codon. And once it arrives at the ribosome, its codon will be exposed here. Fine. So we will search another transfer RNA, another RNA that will be having the same codons, but in that case it will be called as anticodon. In case of the transfer RNA also have triplets of nitrogenous bases, or you can say nucleotides. And in case of the transfer RNA, these will be called as a anticodon. Let's suppose here 1, 2, 3. There are 3, 4, 5, 6, and there will be lot of nucleotides. But 3 nucleotide, a set of 3 nucleotide will be called anticodon. In case of what? In case of transfer RNA, here it will be called as anticodon. So here I mentioned here anticodon. So remember, the sequence of the anticodon and codon must be matching, must be highly matching. Now, how? What if I use the word complementary? That will be more good rather than to use match. No, complementary will be more good, because let's suppose if at the messenger RNA, if there is thymine, let's suppose, okay, my God, thymine is not present in the RNA, so you cannot write thymine here. You can write here uracil. Okay, you can write here uracil, and here is guanine, and here is what? Cytosine. Okay, let's suppose these three bases are present on the messenger RNA. Now, when the transfer RNA come, look, transfer RNA is also supposed to come at the ribosome, and what will happen? The triplet codon, anticodon of the transfer RNA, they will be complementary. Means, if here I write the diagram of the transfer RNA as well. So here, okay, fine. Sorry, we can write it like this. Okay, now suppose this is the transfer RNA. This is a diagram of transfer RNA has arrived, and now you will see that the sequence of transfer RNA and the messenger will be like this. In front of C, transfer RNA must have G, and for G the transfer RNA must have C, and for U it must have A. So we say that the codon of the messenger RNA, what? The codon of the messenger RNA is complementary to the anticodon of the transfer RNA. Once these two RNAs, these two arrive at the ribosome, then after the code matching, one specific amino acid will be added here at the surface of ribosome, and then another transfer RNA will come along with its one amino acid, and similarly for each transfer RNA you will see one small circle that will build like this, and they will make a long chain protein synthesis. So this was something about messenger RNA, and you saw so much discussion of transfer RNA here in messenger RNA as well, because these all are linked together. Now come to the transfer RNA. Transfer RNA's percentage in the cell is 10 to 20%. And one thing more, I just got it now, that messenger RNA length is variable. Its length is variable. So this is more important. Variable means depend upon the protein. If you want a long chain protein, the messenger RNA length will be longer length, and if you want shorter chain, shorter length protein, then the length of the messenger RNA will also be short. Okay, so the messenger RNA length is variable. Okay, it may contain 10 nucleotides, 20 nucleotides, 100, 300 nucleotides, depend upon the protein. In comparison to that messenger RNA, the transfer RNA length is almost constant, fixed. That is 75 to 80 nucleotides. Whatever protein you have, whatever length of protein you have, for this the transfer RNA's length is always constant, that is 75 to 80 nucleotides. And now transfer RNA, what is the function of a transfer RNA? Just like we saw messenger RNA just take the information from the DNA. Okay, the informations are present in what? In the DNA. Let's suppose this is the DNA. Okay, double-stranded DNA, and from that DNA the informations are present. A lot of informations are present, and some of these information will be copied in the form of the messenger RNA. These information will be regarding some protein synthesis. Okay, so the information, a kind of a transcript, has been taken in the form of messenger RNA. Now the transfer RNA will pick a specific amino acid. You know, we know there are 20 different types of amino acids. So there will be 20 different types of transfer RNAs as well. So these transfer RNA, it means each transfer RNA will pick a specific amino acid and it will carry it towards the ribosome, because ribosome is the machinery at which protein synthesis will start, and for protein synthesis amino acids are essential. So who will bring amino acid? Transfer RNA will bring amino acids towards the ribosome. And who will bring the information, the map, the hidden map, that just like you say how to build a bungalow, how to build a room, how to build a house, and for this you first need some sort of map, written map, that that room should contain 2 windows, 1 window, okay? What would be the size of each wall, the length, the width of each wall? All of the information for this one single room is included in a single page. So that information will be derived from the DNA. The information will be from the DNA, and the amino acids will be brought by the transfer RNA. Now, the structure of transfer RNA is important as well. If you see the RNAs, the transfer RNA structure looks like the clove. You might have seen the kind of botanical structure, clove, or kind of a fruit of plant, I think, fruit, yes. Clove, it looks like clove, C-L-O-V-E, clove. It has 5 arms. Look, if it is the 3 end of the messenger of the transfer RNA, then of course it will be the 5 end of the messenger RNA, and a lot of nucleotide bases will be attached here, but we will focus on the arms. There are one arm, 2, 3, 4, and the last one is called the 5th arm. Let's start. This first arm, which you see here, 3 and 5 prime, this arm is called, what is the acceptor arm? And acceptor arm, how many nucleotides are present? How many base pairs, mean just like adenine with thymine, or adenine with uracil, guanine with cytosine, fine. This is called pairs. So how many base pairs are present in the acceptor? There are 7 base pairs are present in the acceptor arm, and remember, acceptor arm attaches the amino acid. What is the function of this? It just bind the amino acid, okay? So then we come to the second, that is called as D arm. D arm contain how many? 4 base pairs, and it helps in the recognition of enzymes. The function of this is that. Then this is the third arm. It is called T5C. Why? Because it contains thymine and cytosine and some a little bit modified nucleotide that we represent with the pi. Okay, so this is not pi, this is psi. This is for psi, P-S-I, psi. So there is not a, you cannot say there is a clear function of that T5C. The most important function is by 2 arms. Number 1, acceptor arm and that anticodon arm. These 2 are very important. The anticodon arm contain 5 base pairs, and these 5 base pairs, among these 5 pairs, 3 are called as anticodon. Let's suppose, as I mentioned here, 3 are called anticodon. And now here comes this small arm is called as variable arm here. The variable arm contains 3 to 20 base pairs, and that variable arm is differ from one transfer RNA to another transfer RNA. You know, we have 20 different types of transfer RNAs. So each transfer RNA will be different from other transfer RNA with respect to that variable arm. So V arm differentiate one transfer RNA from the other transfer RNA. So I think this is also most important, because that is why it is mentioned as V arm. V means variable arm. I can write the full name as a variable arm. Okay, so once again, there are 5 arms. 3 are most important. The first one is the acceptor arm, because it will bind the specific amino acid. The second one was variable arm, that it differentiate one transfer RNA from other transfer RNA, and the third one is in the anticodon arm. This arm is having the nucleotide sequence that will be complementary to the sequence of the messenger RNA. And then we will come in the last we will come to the ribosomal RNA. This is the third type of RNA. Ribosomal RNA is actually the part of a ribosome. Let's suppose this is the diagram of the ribosome, and ribosome contain a smaller unit and a larger unit. A ribosome contain protein and nucleic acid. Protein and nucleic acid. These two molecules, protein and nucleic acid, are collectively called as nucleoprotein. So the whole ribosome, this whole ribosome consists of nucleoprotein, and if you see the nucleic acid in that ribosome, this will be the ribosomal RNA. Means if you find RNA here in that ribosome, okay, so the whole ribosome will contain RNA, of course, but that RNA which is present in the body inside the ribosome is called as the ribosomal RNA. And now the percentage of the ribosomal RNA is from 50 to 80%. Now, what is the function? The function of the ribosomal RNA from the book of Satyanarayan is that its function is not yet clear, and it is hypothesized that the ribosomal RNA helps in protein synthesis. The exact mechanism is not clear, but it helps in some way in protein synthesis. It looks like it helps in the translating the hidden message. What was the hidden message? The message that is coming from the messenger RNA, and that message is always for some specific protein or from for some specific enzyme. So this message will be decoded by the help of what? By the help of that ribosomal RNA. And if we just remove this machinery, ribosome, protein synthesis will stop. Protein synthesis always require some sort of machine, and the ribosome act as a machine, because information will come from the messenger RNA, amino acid will come by the help of transfer RNA, and some sort of machine that will help in translating that message of messenger RNA and these amino acid, that is called as the ribosomal RNA. So this video was about the types and structure of the RNA. I hope you enjoyed watching that video. Thank you. Give us a like and share that video. Thank you. Bye bye.